Okay, good morning. Afternoon now. Welcome everyone to Let's Chat About. This is a free monthly webinar series hosted by us at Hope and Focus. We've developed the series with those living with LCA and IRDs in mind, but it is open to anyone who is interested in what's happening in our communities. Hope and Focus is a patient advocacy organization dedicated to generating awareness, raising funds for research, and providing support, education, outreach to the LCA and rare inherited retinal disease community. My name is Courtney Coates, and I'm the Director of Outreach and Development for Hope and Focus. Before we get started today, I just want to take a moment to thank our sponsors for this series. They are Dominion Energy, Janssen, Mira GTX, and Spark Therapeutics. We could not provide these programs such as these without their support. A few quick housekeeping items. The session will last about an hour. Your microphones have been muted and your cameras are off. You can submit questions through the chat function, and as time allows, we will take any questions that come in live via the chat. Otherwise, we will take all questions at the end of the webinar. We are recording this session, and after the webinar, we will post the recording to our YouTube channel and send out the link. We at Hope and Focus are excited to be sharing this webinar with you today and introducing the community to the Bespoke Gene Therapy Consortium, as well as the 2 inherited retinal disease programs that have been chosen. I am pleased to be joined today by Ben Shaberman, Vice President of Science Communications for the Foundation Fighting Blindness. The Foundation Fighting Blindness is a strong partner to us at Hope and Focus, and Ben serves as one of our advisors as well. Ben will be our moderator for today's discussion, and thank you, Ben, for your help today.
Well, thank you, Courtney. Thanks for all out there joining us, and thanks to our 5 expert panelists who will enlighten us about the Bespoke Gene Therapy Consortium, a really important consortium to boost gene therapy development for a variety of rare diseases, including inherited retinal diseases. And just to give everyone a little context for why this consortium is playing such a critical role in gene therapy development, at least from an IRD standpoint, inherited retinal diseases are rare, but they're an excellent target for gene therapies. And there are a few reasons, but I want to underscore 2. The retina is a great target because it's small, it's easily accessible. You don't need a whole lot of gene therapy to treat it. And just about all inherited retinal diseases, including Leber congenital amaurosis, are monogenic, meaning there's just one mutated gene that's causing the patient's disease and vision loss. And if you can augment or replace that mutated gene, you have an opportunity to potentially save and restore vision. And through the advent of Luxturna, the first FDA-approved gene therapy for the eye or an inherited condition, and many other inherited retinal disease gene therapy trials, we're seeing a lot of promise and success in gene therapy development. But developing gene therapies for any rare disease is challenging, and it's tough in developing gene therapies for inherited retinal diseases, because there are so many genes, each of which, when mutated, can cause an inherited retinal disease. There are about 300. So we need a lot of therapies, ultimately, if we're going to genetically treat these conditions. And about 24 of those are associated with LCA. So there are a lot of technical and economic challenges. But that's where the Bespoke Gene Therapy Consortium comes in. And I'm going to call it the consortium, or BGTC, from here on out. And really the mission or the goal of BGTC is to improve and accelerate development of gene therapies for rare diseases, including those that affect the retina. And without further ado, I want to introduce our first speaker, Brad Garrison. He's a senior project manager of the consortium, and he's with the Foundation for the National Institutes of Health, and he will dig a little deeper into what that consortium is all about. Brad, take it away.
Excellent. Thanks so much, Ben. Did you want me to share the slides, or is that being done
You can
centrally?
go ahead and share if you'd like, Brad.
All right, just give me one moment. Well, thank you so much for the kind introduction, Ben. As Ben correctly mentioned, we do go by the acronym BGTC. That stands for the Bespoke Gene Therapy Consortium. It is one of 7 programs within the FNIH that we call Accelerating Medicines Partnerships. So this is a unique public-private partnership model that helps bring together the top scientists from NIH, industry, as well as nonprofits and patient engagement groups such as Hope and Focus. And so again, I thank you all for having me here today. You can see some of the other key contributors to the team here on the slide. He is co-chaired by PJ Brooks from NCAT, Tim Miller of Thermo Fisher, and Peter Marks of the FDA is also an ex officio co-chair, as the FDA is a partner in this consortium. Program management support provided by Courtney Silverthorne, Kira, and Juan. The contact information is there in the QR codes, as well as a link to our website. If I can advance the slide. To further Ben's point about why we're here and what we're trying to achieve, not only are there 300 monogenic diseases that give rise to inherited retinal diseases, well, in fact, there are over 10,000 diseases that meet the definition of rare. This is defined as affecting less than 200,000 people in the US. And while each individual one might be rare, in their collective, they affect over 30 million people in the US. Obviously, there are equivalent stats that equate this to the worldwide population, but this particular citation is US based. And 80% of those rare diseases are estimated to be caused by a single gene mutation, that therefore are monogenic. Therefore, there is the possibility that we can address these through some form of gene therapy. The BGTC is currently focused on AAV as the vehicle to deliver the missing gene to the affected tissue. But other consortiums are working on different types of gene therapy, such as gene editing or cell editing. The BGTC is organized into 2 major objectives. First of all is a basic AAV biology, in which we're aiming to gain a better understanding of the mechanisms of AAV so that we can enhance vector production and also enhance therapeutic gene expression. So the goal there is to optimize the delivery vehicle, in this case, AAV. The second track, which is probably more relevant to this audience, is the clinical track, where we are going to manufacture the AAV necessary to initiate preclinical testing and ultimately clinical trials for the selected diseases. Along the way, we're going to define best practices in terms of CQA quality testing. So those are the critical quality attribute assays that are done on the manufactured product. And we're also going to look at ways to optimize and streamline the GLP tox testing. All of those learnings and best practices will be coalesced into the BGTC regulatory playbook. The first version of the playbook is actually live today, but it doesn't have the minimum packages yet because they've not been fully vetted. More on that later. Just wanted to quickly highlight the AAV biology research projects that we are funding. 2 are in the area of vector production and area of therapeutic gene expression. The BGTC aims to establish a repeatable, optimized process for clinical development of gene therapies. So in order to be effective in this model, it needs to be addressable. It needs to be a single gene defect of no commercial interest. So the requirements included a short time frame to follow up, because we can't wait years and years to assess the patients, and having an assembled patient group. So having a knowledge of the patient community that's affected. And the goal was to select 5 to 6 diseases with a high likelihood of success to move forward with clinical trials. And again, we would arrange for the manufacturing and clinical characterization and testing of these products. So we went through a disease selection process where we received 62 applications for different rare diseases. The requirements were that there be information about the disease and the disorder, information about the patient demographics, clinical presentations, and clinical and preclinical research history to date. From there, the selection committee narrowed it down to 14 top candidates. And we put out an RFP for clinical trial proposals based on those down-selected diseases. Again, these had to be monogenic disorders with no current commercial business use case and sufficient information to run a successful trial and recruit the necessary patients. From those 14, we selected 8 to move forward with entering into the portfolio of clinical trials. Some of the factors in narrowing it down from 14 to 8 were the cost, the ability to secure AAV manufacturing, follow-up requirements, and diversity of the patient population. We've paired the different diseases with a manufacturer to produce the vector that'll be used in the trial. Here you can see the list of diseases. Obviously, this is more focused on the retinal diseases NPHP5 and CNGB1. Ben mentioned 300 different genes that give rise to inherited retinal disease. Well, I looked up retinitis pigmentosa and found that there's about 100. So this particular one that was selected happens to be number 45 on that list, caused by the gene CNGB1. The way that we're going to manage all of these clinical trials is through the organizational structure you see here. At the top, we have the BGTC steering committee and its associated manufacturing, preclinical, clinical, and regulatory subteams. Each of those subteams has appointees on the cross-functional teams. So as represented in the blue dashed box, there's one cross-functional team for each of the 8 disease indications. In addition to the sub-team reps, we have the sponsor, the PI and their team, who are also here on this call, and the patient advocacy groups have a role on these cross-functional teams. And then there in the maroon box is the coordination center, which consists of the PMO and the 4 different forums: data management, analysis, preclinical, clinical, regulatory affairs, quality assurance, and any CROs that they may hire to conduct the work that is managed out of the coordination center. The BGTC has released, as of a week, or I think it was last week, we released the playbook. So version 1 of the playbook serves as an initial framework. It does not include the minimum CQA and animal toxicology packages that we plan to include in future versions. But as an initial framework, we do welcome feedback from the community. So feel free to scan that QR code and send me an email if you have any questions about the playbook. Again, the idea here is to capture best practices for gene therapy clinical development. And really what it's all about is navigating the regulatory process. We can't change the regulatory process, but we can help guide drug developers on how to best navigate it. And Shout out to our partners. We have multiple NIH agencies, cross-functional initiatives, the FDA, biotech companies, pharma, patient advocacy groups, and other nonprofits. We've raised $100 million so far in this effort. Here's some of the resources that we have available: a video, an animation about AAD, the playbook, and a recent publication in Nature Reviews Drug Discovery. So I encourage you to scan those QR codes and look through some of these materials. And there's my contact information and our website. Thank you all, and look forward to taking any questions. I think we're going to save them for the end. So I think it is now over to the next speaker.
Great. Thanks for that excellent introduction, Brad, to BGTC. And yes, we are going to hopefully have a little time for questions at the end. But to expand on the consortium a little more, I'm glad to introduce Dr. Snehal Naik. She is Senior Director and Head of Regulatory Policy and Intelligence and Regulatory Strategy Leader at Spark Therapeutics. And I'm sure many of you know that Spark is the developer of the gene therapy, the first gene therapy for the eye or the retina, Luxturna. So, Dr. Naik, over to you.
Thank you so much, Ben. Really a pleasure. I appreciate the opportunity to speak with all of you today. I do not have any slides that I'm sharing. Just will share some thoughts briefly based on my participation in BGTC. So as Ben mentioned, I serve as the head of regulatory policy and intelligence at Spark, and Spark is one of the members of the BGTC, and I have had the privilege to sit on the regulatory subcommittee, which is one of the many committees that brings together all of the different members across BGTC. And I think two things that really stand out for me when I think about the BGTC as a consortium, as an opportunity that I think distinguishes it from a lot of other ways that industry partners are normally able to participate. Normally we have these large industry trade associations, or you can have professional societies that also have a collective voice and have the ability to sometimes bring forth to regulatory authorities a more collective industry view and try to shape the field a little bit to make development more efficient or to create opportunities in disease areas where currently there might not be any. I think what makes the BGTC truly unique in my book is twofold. I think one, as Brad showed you the structure, this is an opportunity where there are actually selected disease areas and indications that BGTC as a whole will be helping to move that clinical development forward. And all of us collectively who sit on each of those different committees, we really have the privilege of bringing our expertise, bringing our background, bringing our experience, sharing a little bit of what we have seen work when it comes to some of these the tissue areas specifically. You saw that they're split up with ocular indications, systemic indications, and CNS indications. If you look across the BGTC members, we have varying expertise. We have expertise in manufacturing, we have expertise in each of those therapeutic areas. We of course have folks like myself, or our day job is regulatory and interacting with regulatory authorities. So to collectively be able to bring that wealth of experience and apply it to each of the selected programs to say, if we all work together on this, how can we move the needle, and how can we possibly move it faster than any one investigator would have had to go out and gain each of that information on their own to be able to do that. So I think that is truly a unique opportunity in a consortium context. And I think the other part of this, as Brad also highlighted, is that FDA is very much a member, and really Dr. Peter Marks, this was one of his visions that he had talked about for a while when it came to really making development of therapeutics possible for rare disease indications. And as Ben shared, I think so much opportunity there when we think about inherited retinal diseases specifically. And that's a unique opportunity to then have FDA members at the table when we're having some of these discussions. It will not replace the usual regulatory process. When we think of developing these, those investigators and those teams will still need to have their appropriate interactions with FDA to make sure that they're moving forward in a way that FDA finds acceptable, and of course get that FDA feedback to help shape that future program. But I think it really is a tremendous advantage when we think about things like the regulatory playbook, where you know that you've had FDA members weigh in on the different committees and put that content together in a way that really reflects their current thinking and reflects their, I think, commitment to this partnership of really saying, how can we bring manufacturers, biotech, academic investigators, and regulators together rather than waiting until things get to a certain stage and then start to put them in front of regulators. So I think this overall ability to, I think, streamline development is truly valuable, I would say, across the board. Valuable, of course, to patient communities, to whom the therapies couldn't come any sooner. But I think equally valuable for all of the other members of the consortium as well, to then take those learnings and apply it, hopefully, to the next iteration and the next iteration, and expand our reach. So Ben, thank you for the opportunity, and I look forward to hearing about the two specific programs.
Thanks, Snehal. And Brad and Snehal, thanks for that introduction to BGTC. And it makes me think of the adage, a high tide lifts all boats. And in a way, the BGTC is that high tide. And it really has an opportunity to boost the entire gene therapy development industry, especially for rare diseases. Okay, and yes, I'm excited to hear about two projects that are underway through the BGTC for inherited retinal diseases. And next, two people I've known for a long time, and I really respect and appreciate their great work in the inherited retinal disease space, are Dr. Artur Cideciyan and Dr. Tomas Aleman. They're both working on the development of a gene therapy for LCA caused by mutations in NPHP5. So first, let me officially introduce Dr. Cideciyan. He's a research professor of ophthalmology and co-director of the Center for Hereditary Retinal Degenerations at the University of Pennsylvania. And he is the go-to person when it comes to imaging and functional testing for the retina. So Art, take it away.
Well, thank you very much, Ben, for that introduction. And thank you, Laura and Courtney and Hope in Focus for this opportunity to reach out and interact with the community of patients and families and caregivers. And of course, our mission at the Center for Hereditary Retinal Degenerations in Philadelphia is to provide basically better sight, either today, tomorrow, or in the future for people living with inherited retinal diseases. We do this by clinical care and research, both clinical as well as translational, and we perform clinical trials. Indeed, we have 5 active clinical trials right now. So for us as being interested in inherited retinal diseases, the key cell that we look at are the photoreceptors in the retina. And on this slide on the right is a schematic of one little segment or portion of a photoreceptor cell. And these are very long cells, elongated cells. And one tippy tip is basically an antenna that catches light and starts the signaling process of vision. And it's called the outer segment, which is shown on the upper right part here on the slide. And the outer segment, for it to function, requires all its proteins to come from a neighboring compartment called the inner segment, where all the energy is for the photoreceptor, as well as where all the proteins are being made. And it turns out, very relevant to the diseases that I like to talk about, is this tiny little isthmus between the inner segment compartment and the outer segment compartment, as this tiny region is called the connecting cilium or ciliary transition zone. And even though it's small, a very large number of proteins are specifically located there and presumably do very important things, much of which is not perfectly understood. And among these ciliary proteins, two of which are important for us today. One is called CEP290, also called, the same protein is also called MPHB6. The other one is called IQCB1, and that's also another name for it is MPHB5. These two proteins form, we believe, a multi-protein complex that locate in here and in the transition zone. And when they are mutated individually, they cause severe problems with vision. And so what exactly is the retinal hallmarks of disease resulting from either one of these two proteins, which turn out to be basically what in the business we call phenocopies, meaning they copy each other. They're very similar. And here I'm showing an example of a cross-section of the retina in a healthy eye, and highlighted is a blue area, which is where all the photoreceptor nuclei are. So when the photoreceptors are alive, they have nuclei by definition, and the nuclei would be located in this region, and by its thickness, we know whether they're alive photoreceptors or not, or if the proportion of them are surviving. In there are both rod photoreceptors as well as cone photoreceptors. Cones are for daylight and rods are for night vision, but they're not just randomly thrown around. They are distributed in a very specific fashion, in the sense that in the center of our vision, in a healthy vision and healthy eye, there are all cones for day vision, and they slowly get less of them as we move away, and there's more rods as we move away from the center. When we look at the cross-section of retinas with people who have either CEP290 mutations or MPHB5 mutations, what we see is only a central mound of photoreceptors remaining, highlighted here with the blue on the bottom. And those are, we believe, retained cone photoreceptors. However, they're not functioning appropriate to the amount that they are there. So they're dysfunctioning but retain cone photoreceptors that tend to stay alive and for decades, and thus opening up a window of therapy to try to treat them. So many, many years ago, we predicted that because CEP290 and MPHB5 mutations result in this retained cone photoreceptors that lack vision, all one had to do was a gene-directed therapy to undo the defect, and vision would recover. Of course, that was just a hypothesis, just a thought, and but it turned out we had opportunities to test that in CEP290 form of the disease in human trials. There has been two trials. There is gene editing trial. It was a multi-center trial for CEP290 disease. One of the centers was at Scheie Eye Institute in the Inherited Retinal Disease Clinic at our place. And the other clinical trial was doing splice modulation using something called cepofarsen, which is an antisense oligonucleotide, which was also a multi-center trial. And we were involved heavily in designing and performing that trial. The results of the gene editing trial are not publicly released yet, but hopefully soon. But the results of the splice modulation version have been extensively published between about 2019 and 2023. And what they showed is in a subset of patients, there is dramatic and clear evidence of vision improvement in a short period of time upon these gene-directed therapy, thus at least superficially confirming the hypothesis that if one can undo this genetic defect, vision should return. It turns out the evidence is more than just a human disease. We have been for decades working with our friends and colleagues, Gus Aguirre and William Beltran at the Veterinary School at the University of Pennsylvania, who have spent their lives looking at naturally occurring blindnesses in dogs. And one of those is caused by, it turns out to be by MPHP5 mutations. And quite amazingly, these dogs have very severe visual dysfunction. And very early in life, they have no visual signals, which are shown on the right with the flat wiggles, as opposed to a healthy control eyes, which show bigger wiggles. And this lack of function, lack of vision in these dogs is not associated with as much loss of cells, meaning there is a lot of cells that are not dysfunctioning. And indeed, at certain stages of the disease, such as shown on the right, the features of the dog retina are very similar, almost copying the human retina with the retained central cones that are dysfunctioning. And thus, one could perform, and indeed, our colleagues and us performed gene therapies in dogs for the MPHP5 mutation. And the results, this is a complex slide, but basically it shows that if you don't inject it, there is a little bit of photoreceptors in the central part. But if one injects right here gene therapy, there is a large segment around the injection where photoreceptors are retained, and they're not only retained, but they also function as shown at the bottom. And furthermore, the results are not just at the level of the retina, but they reach the brain. These are recent results from dogs that have been unilateral gene therapy, where the brain images when the treated eye is evaluated show a lighting up of the brain, implying that the dogs are in their brain perceiving vision. Whereas when one stimulates the untreated eye, there is no such thing. So then in summary, CEP290 and MPHP5 proteins are components of a multimolecular complex at this very important ciliary transition zone portion of the photoreceptors. There's comparable, almost copied retinal disease resulting in CEP290 mutations or MPHP5 mutations in humans, as well as MPHP5 mutations in dogs. Common across human and canine retinas are photoreceptors that are dysfunctioning, thus opening the door for molecularly switching them on. And human clinical trials in CEP290 do show improvement of cone vision in some of the patients. And canine gene therapy also shows improvement of vision as well as preservation of photoreceptors, thus bringing me to the conclusion that gene therapy for the human retinal disease caused by MPHP5 has great potential. And we are really excited to be part of the BGTC consortium to try to lead up to evaluating this in a gene therapy. Thank you very much.
Really nice review, Art, of NPHP5, the canine studies, and how all that you've learned over many years provides a lot of hope that gene therapy for NPHP5 in humans has exciting potential. Dr. Aleman, you are up next, and let me give our listeners and viewers a little overview of who you are. You are the Irene Hines Given and John Laporte Research Professor and Director of the Retinal Structure and Function Laboratory at the University of Pennsylvania. And perhaps most importantly, you provide great care and analysis and testing for people with inherited retinal diseases. And I know you're a PI on clinical trials for emerging therapies. So
Ben, the plan was basically Art would give the talk for both of us.
Oh, okay.
And I was going to piggyback. Of course, I'm here also as a clinician for whatever questions that may come from the audience. But we figured that one of us could do for both of us.
Okay.
So I think we pass the baton to the next.
Okay, well, thanks for joining us, and that will leave us potentially more time for a little Q&A. Thank you for being here, Tomas. So finally, to wrap things up, we're going to switch gears a little bit and learn about an emerging gene therapy for retinitis pigmentosa caused by mutations in the gene CNGB1. And I'm pleased to introduce Dr. Aykut Demirkol. He's an ophthalmologist, a surgeon, and assistant professor at Üsküdar University in Turkey. I hope I said Üsküdar correctly. And he's also doing some great postdoctoral research at Columbia University. So, Dr. Demirkol.
Yes. Thank you for your kind introduction and opportunity to speak at this meeting. Can I share my screen?
Please do.
Just a second. Yes. Okay, so today I'm working in postdoc researcher in the Columbia University Department of Ophthalmology. So today I want to talk about our CNGB1 project. Before that, I want to talk about the CNGB1. So mutation in the CNGB1 gene profoundly impacted the photoreceptor cells within the retina, leading to the retinitis pigmentosa, a condition marked by a progressive vision loss. In the simplest term, the CNGB1 gene, when it doesn't work right, causes a type of vision loss called autosomal recessive retinitis pigmentosa, which is a big name for a condition where people lose their sight over time. It starts with not being able to see well at night, and then moves on to the losing more and more of the sight vision. Kind of like looking through a tunnel that gets narrower and narrower. It can even lead to a person seeing speckles, and also this usually happens to folks when they are in their late 20s and maybe 30s. The progression of retinitis pigmentosa due to CNGB1 mutations underscores a critical and unmet need for effective treatments within the ophthalmological landscape. Current therapeutic strategies are largely palliative, focusing on symptoms management and slowing the disease progression rather than offering a cure or reversal of the condition's course. So the concept of the gene therapy represents a groundbreaking advance in medicine, especially for inherited retinal disease or inherited other disease like retinitis pigmentosa caused by CNGB1 mutation. It's like having a superhero in the medical world. Instead of relying on chemicals, gene therapy uses a virus to deliver a healthy CNGB1 gene directly to the retina. This superhero approach doesn't just aim to slow down the disease. It holds the potential to reverse the same of the damage or offering hope for improving vision. But gene therapy isn't just limited to one problem. It's like, what can I say, unlocking a treasure trove of possibilities for treating various type of genetic eye disease and bringing us closer to personalized medicine. This breakthrough highlights the incredible potential of genetic research and technology to transform patient care and enhance the quality of the life. So this is in our preclinical studies involving mice, dogs, and non-human primates. We have seen promising results with AAV5 mediated gene replacement therapy for CNGB1 retinopathy. This therapy has shown potential to repair and preserve the structure and function of the retina, which could be a significant step in treating this kind of eye disease. So in the clinical study, adults with autosomal recessive retinitis pigmentosa due to specific mutation in CNGB1 gene are given a gene therapy through an injection into the eye. We believe this therapy, AAV5, will be safe and will help maintain a critical region in the eye responsible for the vision, known as the ellipsoid zone, without causing a significant safety concern. The endpoints spanning safety evaluations, visual function assessment, and genetic expression analysis are comprehensive and designed to capture a holistic picture of the treatment impact. So in this intervention, being studies involves the use of vector called AAV5 to deliver a healthy copy of CNGB1 gene into the eye via a subretinal injection. This vector acts as a delivery vehicle carrying the correct version of the CNGB gene into the cells of the eye. The CNGB gene is essential for producing a protein that plays a critical role in the proper function of the photoreceptors cells in the eye. So before treatment, the rods in the retina may be unhealthy due to disease. The injection introduced the correct gene into the retina, aiming to express the right protein to restore the rods for healthy condition. After treatment, the goal is to have a healthy rods and outer segments, potentially improving or preserving the patient vision. The AAV5 factor is paired with a human rhodopsin promoter, which acts like a biological on switch directing to gene to work specifically in the right cells of the eye. So further changing the precision and effectiveness of the treatment. In this clinical trial, focuses on evaluating the safety and effectiveness of AAV5 gene therapy for retinitis pigmentosa caused by the CNGB1. This initiative treatment approach involves delivering the therapy directly to the retina through the subretinal injection. The primary goal of the study or primary endpoints are check for safety and how well participants can handle the treatment, known as the tolerability. The second goal or secondary endpoint is to see if the treatment is effective or effective and specifically looking at whether it can keep a critical part of the eye cells called ellipsoid zone stable in their size. This image shows the outlines of the structure of the clinical trial. Participants undergo a treatment screening, pre-treatment screening, followed by the treatment period with various check-ins and then a follow-up phase for further assessment. The trial is carefully monitored by a safety committee, and data collection during the study will be analyzed to assess the safety and efficacy of the treatment. This study is controlled trial where participants are divided in 3 groups. One group gets a lower dose, the other gets a higher one to see how well they tolerate it and how effective it is over a period of 36 months, and also a control group with no injection. This approach helps us to find best dose that's both safe and effective for future treatments. So the clinical study is being conducted in 3 sites. One of them is Columbia University in New York, and the other 2 is international sites in Germany. One of them is Munich, the other one is Tübingen. These locations are also significant because they are already working with the patients who has CNGB1 retinopathy. They have been involved in a natural history study of the conditions. So this means they have been tracking how the disease progress over the time in patients. So this peer experience is valuable for the upcoming gene therapies or gene therapy trials. Success of the treatment, gene treatment could be significant impact medical research because current treatment just try to ease the symptoms because we don't have a cure yet. That's why we are super excited about gene therapy, which might fix the problem right at the source. So you can fix the problem at the source, the wrong gene. This could be a huge progress because it might stop the disease from getting worse or even improve sight, which would be a treatment for this kind of a eye problem or maybe more. So thank you for this meeting and the opportunity of the speaking in this meeting. Thank you.
Thank you, Aykut, for that really nice overview of CNGB1 gene therapy. Very exciting. It sounds like you're ready to start dosing patients, or have you dosed patients yet?
We are just a natural history part. So we are enrolling the patients, already enrolled the patients, and we are follow up these patients in clinical sites.
Got it. Got it. Thanks again. So we have time for questions, and we've gotten a number of good questions. And by far the most common question is about the emerging NPHP5 gene therapy. And I'll ask it in two parts. What else needs to be done to move that gene therapy into a human trial? And I know this is always a tricky question, but when do you think that clinical trial may actually begin? And obviously, I'll pose that to Dr. Cideciyan and Alaman.
I'll try to answer that. So the obvious first step is the production of a clinical grade vector. So this is the gene therapy material that's going to go into human eyes, and then once that material is produced, that needs to be tested for efficacy and toxicity in a new set of preclinical, meaning animal studies. And the results of all of that and the information about manufacturing the clinical material will be submitted in what's called an IND to the FDA. And once the FDA gives approval, clinical trial can start the next day, assuming everything else is in order. Thomas, anything to add?
No, that we have done the background work on the clinical side of things to have the proper readouts ready. We have the experience with the CEP290, which, as you suggested, is almost a exact copy. So we think that on that side of things that we are ready to go, basically, once these type of things get sorted out.
Thank you. Thank you. And I want to emphasize, if you agree, Tomas and Art, that manufacturing is not a trivial thing. To manufacture the vectors with the quality for use in humans, you really want to make sure they're safe and they're the right formulation. That's a pretty big deal, and that's one of the goals of the BGTC is to really come up with best practices for manufacturing. So we've gotten a couple questions about the consortium. So Brad and Snehal, the first question actually that I have, and I think would be an interesting question, and it relates to one of the questions from one of our participants, is how much is being invested in the BGTC, and where does that money come from?
Well, that's a great question. And the answer is we've raised $100 million so far for the 8 projects. And that money comes from a combination of donations from the NIH as well as our private sector partners. So if you remember that slide with all the logos on it, they all contributed funding, or in some cases in-kind contributions. So some of the manufacturing is actually going to be donated to the consortium as an in-kind service. So that's how we are funding these trials.
That's really cool that you've gotten all these different both commercial entities and nonprofits to contribute. And just to emphasize, any funds provided by the NIH are taxpayer funds too. So it's your tax dollars at work, if I'm correct. And Brad, you answered this question in the chat, but I wanted to have you verbalize it as well. As the consortium moves forward and you reach milestones and advancements, how are you going to communicate those?
Yeah, well, the best way to stay informed of consortium activities and milestones is to sign up to our newsletter. We regularly send out communications around major milestones and things like that. And we also try to have a presence at the various gene therapy conferences. We are partnered with ASGCT, and every year at their annual meeting, the BGTC annual meeting will also be embedded. So this year it's Wednesday, May the 8th from 1:30 to 5:30 p.m. in Baltimore. So join us there if you'd like to get some live updates. We'll be presenting the playbook and accepting user feedback live. So that would be an opportunity to provide feedback on the playbook itself and ask questions of the consortium leadership.
Cool. And Brad, what is the URL people can visit to just learn more about the consortium and maybe see news items?
Yeah, it's fnih.org/BGTC.
Okay. Say that one more time, just so people get it.
Yeah, fnih.org/BGTC.
Right. And if you Google it, like I did in preparation for this webinar, you can easily find it. And I'll add that we at the foundation will report on some of the advancements, especially for NPHP5 and CNGB1 as they are made. And I don't know if we really have other.
There's some new questions that just came in, Ben.
Are there?
Yes.
In the Q&A?
Yes, in the Q&A on the open.
Okay. And this is a good question to clarify for Art. So you mentioned there are 5 clinical trials underway for gene therapies. Can you say what those trials are targeting?
Yes, happy to. So these are 5 clinical trials that we are running at the Center for Hereditary Retinal Degenerations. There is a lot more across the world. So in our center, there's 5 active ones. One is for LCA1, which is guanylate cyclase form of Leber-congenital amaurosis. And it's fully enrolled, and we are following those patients. The second one is LCA5, which is the first cohort has been injected, and preliminary results will be presented at the ARVO in 2 months. The third one is the gene editing for LCA10 or receptor 90. And that was also fully enrolled, and the results are hopefully going to be published sometime soon. And we are following those patients. And the 4th gene therapy trial is retinoschisis, X-linked retinoschisis due to RS1 mutations. And that is also a multi-center trial that just recently started, and we are part of that. And the fifth clinical trial that's active is not gene-based, it's gene agnostic trial. And as a NAC, it's an oral medication in a phase 3 trial for patients with retinitis pigmentosa of any genotype, but they need to have certain retinal features to be able to be part of that.
Great. Thanks for the overview of the 5 trials underway at Penn. And I took the liberty of putting a link to a document we maintain on the Foundation's website of all clinical trials that are underway. So obviously people can click on that. And we are just about at the top of the hour. This has been a really comprehensive and eloquently presented overview of BGTC and some exciting gene therapies for NPHP5 and CNGB1. Thank you to all our panelists and everyone who tuned in. And Courtney, do you have any final words before we sign off?
Yes, thank you so much, Ben, for being our moderator today. I'm just going to note there are some additional questions still in the chat that we are running out of time to get to today. If you'd like to email us directly at info@hopeandfocus.org, we will do our best to address the questions that we are able to offline after the webinar. Additionally, I'd just like to say thank you to everyone today for joining us. This has been an incredibly informational webinar, and we look forward to seeing your programs advance through the BGTC model. And we look forward to hopefully having some additional webinars in the future as these programs expand and we get into more details. So to stay up to date with everything that we have going on here at Hope and Focus, please make sure to sign up for our emails and our newsletters so that you can continue to stay informed. We will be dropping a link in the chat. If you would like to support the work we are doing, there is also a button to donate in the top right corner of the website. And lastly, we always recommend that you check out Ben's Eye on the Cure podcast. He also hosts important conversations on inherited retinal disease. We will be dropping a link in the chat, but highlights a bunch of different speakers and is always a great time as well. As always, stay tuned for our next webinar. We look forward to talking with everyone again soon.
Thanks, Courtney and Laura. Great work at Hope and Focus.
Okay.